And by the way, right now we're going to talk about AAV vectors again with Anne Bello. You can join me. Thank you. You will talk about next generation AAV vectors for gene therapy and muscle disorders.
Okay, so good morning, and first I would like to thank the organizers for giving me the opportunity to present some of the work that we are doing in our laboratory, and I would like as well to thank Dr. Ajali for the very nice and comprehensive introduction on the AAVs and the technology itself. So I am going to show you just a few examples of, very concrete examples of some of the work that we are doing at the technological level in our laboratory. And so if I will try to have the slides. So, as we know, gene therapy has changed the therapeutic landscape in medicine. And our laboratory, which has been has performed pioneering research in this field with the development of gene therapy products that are in clinical trials. We are particularly interested in developing and finding new treatments for neuromuscular diseases, which account for more than 400 different diseases and altogether affect more than 1,100 people in France. So, so far, a few AAV gene therapy products have been approved, well, in fact 6, as Omeya was saying just before, and only 2 of them are approved for neuromuscular diseases. One for spinal muscular atrophy, which was approved in 2019, Zolgen-SMA, and another one which was approved last year for Duchenne muscular dystrophy, which is Elevidys. So these gene therapy products are using AAV vectors that are derived from natural serotypes. And these serotypes have some limitations, as was mentioned just before by Dr. Ajali. So there are many challenges still for gene therapy, and in particular, for example, for the use of high vector doses in order to achieve therapeutic efficacy, and in particular in neuromuscular diseases, but as well, for example, the high seroprevalence of neutralizing antibodies in the human population, which prevents some patients to be treated. As well, for example, the high manufacturing costs, which is as well a main challenge. So these limitations have created a high demand for new synthetic and improved AAV vectors that are more efficacious, that are more specific, that are safer, and that have a remaining capability to be produced at large scale and at the clinical level. So in our lab, we are trying to develop new AAV vectors with an increased potency for skeletal muscle in order to treat muscle diseases and establish proof of concept in animal models for rare diseases, in particular for muscular dystrophies, congenital myopathies, but also other myopathies. And we are using different approaches, many of them as were mentioned just before by Dr. Ajali. So we are, for that, we are using several approaches, as indicated here. We are using capsid shuffling. We are using as well computational rational design and artificial intelligence in order to generate new capsids with improved properties, in particular an increased tissue tropism, a low seroprevalence in the human of neutralizing antibodies in the human population, but as well to increase manufacturing yields. And I'm going to give you a couple of examples of some new myotropic AAV capsids that have been developed in our lab or in collaboration with other teams. So the first ones are the AAV Myo2 and Myo3 capsids. So these capsids were developed in the context of a European consortium. And the technology that was used here is the DNA shuffling library strategy, so in vivo screening of capsid libraries in order to select the best capsids. And in addition, there was a peptide that was inserted in a specific region of the capsid in order to increase tissue specificity. So by doing this, we were able to select 2 capsids that were tested in animal models of muscle diseases, in particular X-linked myotubular myopathy and Duchenne muscular dystrophy. And we were able to show that intravenous administration of vectors composed of these capsids were much more potent. So we were able to show that by decreasing the doses by about 10 to 50 times of what is used in clinical trials, currently in clinical trials, we were reaching clinical efficacy in these animal models. And as you can see here, by looking at the molecular level, the expression levels of the mRNA derived from these expression cassettes, we found that there were 10 to 100 more mRNA derived from the transgene than when we were using a controlled natural vector, so an AV8. In addition, this capsid was able to as well detarget AAV vectors from liver transduction, which is particularly interesting when we are trying to treat muscle diseases in which we don't need to target this organ. So this can reduce potential side effects in this organ. As another example, I'm going to show you another capsid that has been developed in our laboratory. This was done by combining computational rational design with artificial intelligence using a bottom-up approach. So basically what was done is we were able to generate artificially new capsids, and in particular we're able to design a specific peptide that could be inserted in a specific region of a capsid that had some nice properties. In particular, this capsid was already able to detarget the liver. And by doing that, several capsids were generated and tested in vitro and in vivo. In vitro, for example, for manufacturing yields, but as well for transduction properties in specific cell types. And these capsids were able as well to improve transduction efficacy in specific models. And in particular, I can show you here, among these capsids, there was one that was the best, which is called the LICA1 capsid, that shows a high transduction efficacy in skeletal muscles and in heart. You can see here on these images of bioluminescence that is indicating the expression of luciferase activity. You can see that there is more luminescence in muscles and heart in these. Here you can see in mice that have been treated with this capsid as compared to a parental capsid that do not contain these, for example, this peptide, this specific peptide, or a natural AV9 serotype. So by administrating this vector in several animal models, we were able to show that this capsid leads to better efficacy at lower doses, so in the range of 5 to 10 to 12 VGs per kilo, we are able to see a therapeutic effect, so that's much lower than what is seen in clinical trials. And in addition, this capsid can as well detarget the liver. So it is as well a very promising capsid that was developed in our unit in the team of Dr. Richard. So altogether, I have shown you that we can use novel technologies in order to design and synthesize novel AAV capsids with improved properties, and in particular, increased potency and specificity in skeletal muscles in order to decrease vector doses and manufacturing costs. In our laboratory, we have as well a long-standing expertise in vector manufacturing, and we and in the optimization of bioprocesses in order to produce a vector and improve the production of vectors at small and large scale. We have as well the capabilities to perform multi-level analysis at the preclinical level in several animal models and establish proof of concept studies with products that can be translated in clinical trials in patients. So this is to show an example of what we can do in our laboratory, which is the unit, the INSERM unit 951, which is an UMR unit located in Geneton at Évry. And we have been able to work on, as I mentioned before, to work and develop gene therapy products that are now in clinic with very promising results. So we can discuss more about that if you want later on in the B2B sessions. I will be very happy to share more information about the work that we are doing in our lab and the capabilities that are available in our lab. And I thank you very much for your attention.
Thank you very much, Anne Bello. And of course, B2B sessions, but you will also have the opportunity to ask directly your questions in a few minutes during the question and answer session after the two next short talks.